Our lab is interested in the analysis of how non-coding RNAs or riboregulators can control root plasticity. What are non-coding RNAs? In fact, non-coding RNAs are the major part of the eukaryotic genome. We can see here, for example, that in prokaryotes there is around less than 25% of the non-coding of the genome is non-coding DNA, whereas when we start to sequence the eukaryotes, still plants and animals, the large majority of the genome is non-coding. However, what is also more interesting in the last years is that there are many long non-coding RNAs who can act either as inhibitors of the microRNAs, but also can have a wide variety of functions regulating splicing, regulating epigenetic pattern, epigenetic regulation, or regulating through antisense RNA the expression of the genes. The model we use in the lab is root developmental plasticity. We can see here that just by changing one mineral nutrient in the soil, we see a total drastically different way in which the roots are growing in the soil. And this adaptation of the roots to the environment is very important because what allows roots to really get all the nutrients they need, or the water, or other components present in the soil. In our lab, we use this phenomenon of root developmental plasticity as a model to try to understand how non-coding RNAs can impact gene expression. The idea we try to support is that the environment, either through abiotic stresses or because of the presence of microorganisms or fungi or pathogens, will change the expression of these non-coding RNAs and in this way affect the regulatory genes. And this will mean that somehow the lateral root organs will change and we will lead to developmental adaptation. Basically follow two lines in our laboratory. A first line is to try to link non-coding RNAs to root adaptation by exploring ecotype diversity. In a second aspect that we are trying to adapt in the lab is to try to understand how developmental mechanisms involving non-coding RNAs can regulate root gene expression. So for example, Thomas in our lab is comparing different ecotypes of Arabidopsis and try to see what are the non-coding RNAs linked to each of them and how they may regulate the ways in which roots grow in the soil.
Hi, I'm Thomas. So I'm interested to know how the non-coding RNA are influencing the adaptation of the different accessions to the environment. Taking phosphate starvation as an example and using RNA sequencing, we are able to show that the coding part of the genomes of the two accessions are almost the same, while the non-coding part is very, very different. So our next challenge is to understand how the non-coding RNA may influence the root growth.
Thank you, Thomas. As we have seen, Thomas is applying genomics to try to understand how are ecotype-specific non-coding RNAs linked to different root growths in different ecotypes. But a second aspect that interests us a lot are the developmental mechanisms by which non-coding RNAs can regulate gene expression and affect the important expression of these regulatory genes.
Hello, I'm Aurélie. So one of the critical events to integrate the function of non-coding RNAs and their target in root growth response is to define precisely where they are expressed. In our lab, I'm interested in assessing this question using hybridization in situ techniques, reporter gene fusion, and microscopical approaches. So, for example, in this picture, you can see the activity of the Apollo promoter on the basis of emerged lateral roots.
Hi, I'm Federico. Actually, together with all the cell biology approaches, we've performed a wide range of chromatin-related techniques in order to try to understand how long non-coding RNAs and protein partners can regulate gene expressions in plants. In particular, we have studied a long non-coding RNA from Arabidopsis called Apollo, who can regulate the formation of a chromatin loop encompassing the promoter region of its neighboring gene, B. Strikingly, this long non-coding RNA turned out to be differentially expressed across ecotypes.
Thank you, Fede and Aurélie. One critical question is, all these genes non-coding? Or they may be coding? And this is a very interesting question because we can imagine that they are also coding for very small peptides on which the computer programs you are using to detect them are not efficient.
Hi, I'm Jeremy. Another critical question about non-coding RNA is to understand their localization in the cell and to know if they can actually encode for small peptides. Hence, I'm interested in the association of this molecule with ribosomal complexes in the cell and to understand their regulation.
Thank you, Jeremy. One last aspect that is being explored in the lab is how other plants than Arabidopsis, Medicago truncatula, which has the capacity to enter in symbiosis with organisms, may also use non-coding RNAs for the normal organogenesis.
Hello, my name is Jeremy. I'm working on Medicago truncatula. In our laboratory, an important aspect is to know if the biogenesis of RNAs can be modulated during development to control epigenetic regulation. Our goal is to understand how RNAs can have an impact on the symbiosis between Medicago truncatula and rhizobia.
Well, as you have seen, using multiple approaches, we try to understand how non-coding RNAs can regulate gene expression with two aims. On one hand, provide new strategies that may help us to modulate root architecture and to bring new tools for controlling how roots grow in the soil using non-coding RNAs. But on a second hand, we hope that these non-coding RNAs will become also tools to be able to modulate gene expression through splicing, chromatin regulation, and hence provide new tools that could have wider biotechnological roles.